we've seen the challenges that avian influenza represents to poultry in the recent outbreaks in Victoria and New South Wales and the ACT. And then on top of that, we have this risk of H5N1 coming into Australia, where if that got in, it would be potentially devastating to our poultry industry. So this is really saying we recognise that avian influenza is a clear threat and we want to do something to intervene. So what we do in this project is we've set up a lab-based system to study the evolution of avian influenza. So what we do is we take cells from chickens and we grow them in the lab, and then we infect them with the avian influenza virus. And we do this successively, so we give time for the virus to evolve. And then what we can do is we can actually modify the cells and look at, if we modify this host factor in the chicken or this host factor in the chicken, can we change the evolution of the virus? And ideally, can we reduce the mutations that we know are associated with increased severity in chickens, as well as potentially transmission to other species? Some of the major findings of our research so far is really focused around this protein that's expressed by chicken cells called neuropilin. And what we found is that when we look at neuropilin levels in chickens and ducks, they're roughly the same. But what's very different is that ducks have this inhibitor of neuropilin that chickens really have low levels of. So that's telling us already that there's something interesting between species, and that's particularly interesting for us because we know that the virus evolves more in chickens than it does in ducks. So then what we can do is we can actually take synthetic versions of that inhibitor. We can force the virus to evolve either in the presence of that synthetic inhibitor or in the absence. And so far what we're seeing is really, really interesting, that the presence of that synthetic inhibitor is actually reducing the evolution of the virus, in particular some of the key mutations that we know are associated with making the virus worse. So that's a really exciting indicator that maybe we have a way that we can intervene with the virus evolution and actually reduce the severity of avian influenza. The next steps of this project would be really moving it from the lab to an in vivo setting where we could start testing in chickens. So what would be ideal, if all the results are very promising, is we could actually start treating chickens with some of these inhibitors, the proteins that we think are driving the evolution of the virus, and say, okay, if these chickens get infected with the virus, does the virus evolve slower? Do the chickens do better compared to a regular chicken? And could we start looking at adding some of these to either the feed of the chickens or selective breeding of the chickens to increase the expression of some of these proteins? So it would really be moving from a lab setting to real life in vivo experiments.